And this leads us on discussing the transport around the cell. So, as you know, there are different kinds of cellular membranes. You have the plasma membrane, the rough ER, the smooth endoplasmic reticulum, the Golgi apparatus, mitochondria, the nucleus, secretory vesicles, lysosomes, peroxisomes, endosomes, and whatnot. And each cell is composed by different fractions of these different kinds of organelles, which are nothing that different compartments bordered by plasma membranes. And so the lumen of vesicles, the lumen of the ER, the lumen of the Golgi, think about it, it's in continuity with the outside of the cell. Okay, so it's like the cell brings the outside inside in the lumen of these. This is very important because the cytosol of a cell is a reductive environment, while the outside of a cell is an oxidative environment. And this will change a lot the nature of the chemical components of what you have in the cytosol versus what you have in the lumen or on the outside of a cell. So, as we already mentioned, cells are compartmentalized. You do have the nucleus and the plasma membrane, you have the mitochondria, you have the Golgi system, you have the endoplasmic reticulum, you have the endosomes, lysosomes, autophagosomes, peroxisomes, and finally actin filaments, which are not plasma membranes, but they're filaments of a protein called actin, which is very important for the shape, motion of a cell, but also for the movement of substances across the cell. You have microtubules, which, analogously are part of the cytoskeleton of the cell, but they're made out of alpha and beta tubulin. And you can see how much of a mess these can be. Yeah, you have the endoplasmic reticulum in yellow, the membrane-bound ribosomes in blue, free ribosomes in orange, microtubules in bright green, dense core vesicles in bright blue, clathrin negative vesicles in white, clathrin positive compartments and vesicles in bright red, clathrin negative compartments and vesicles in purple, and da. So it is fundamental to know how things move from one compartment to the other. And one very important concept you will always have to bring with you along your whole life is the concept of signal sequence. Signal sequence is a sequence within the protein and usually on the N term of the protein, which directs the protein into the different compartments of the cell. So here, for example, you see the Lis, Arg, Lis sequence, which will direct the protein towards the nucleus, export from the nucleus, import into mitochondria, and so on and so forth. You don't have to know them by memory, of course, but it is important that you do know that they are there, that the movement between the different compartments of the cell of a protein is not happening by random, but is directed by these sequences. So the signal sequences, occur as, part of the translation of the protein. And as we said, are on the N term of the protein. But other than the signal sequences, there are other factors that will help the cell to know where to direct that protein, such as post-translational modifications, which are modifications which occur after the protein has been, after the mRNA has been translated into a protein. And it's a non-aminoacidic, a non-amidic covalent alteration of the structure of the protein, which will result in a change in the function of the protein. And this can be, for example, glycosylation, which occurs in the Golgi system, and it will consist into attaching a glycoside or sugar to the protein to attach it covalently with a covalent bound. Phosphorylation. I'm pretty sure you're pretty familiar with phosphorylation. A phosphate, a PO3, will be added via condensation to specific residues in the protein. This can be usually serine, threonine, and tyrosine. And this will also direct the proteins into different compartments of the cell. And then, of course, the last but not least, ubiquitination, where a protein called ubiquitin will be added on the C term of a protein, and it will direct it to this machine, the proteasome, which is involved in the controlled degradation of proteins. So, to sum up, there are three main ways the proteins are transported around the cell. Gated transport through the nuclear pore, the transmembrane transport through the mitochondria, and the vesicular transport through the motor proteins. And I will explain these in the next section.